Hi, I'm Dr. Meredith Barr, and I'm going to be talking about some work we've been doing in my lab related to regulating the concentrations of nutrients in soil using a combination of plant and animal waste biochars. According to the House of Commons Environmental Audit Committee in the UK, agricultural pollution is the most common driver of ecological damage to UK waterways. And that's why the UK government introduced a legally binding target to reduce nitrogen and phosphorus loads from agriculture by 40% by 2038. So the mechanism of this is that when fertilizers are applied to agricultural land, if there are excesses that aren't used by plants, they can enter waterways via runoff. This causes algal blooms to occur in these waterways, which shades submerged plants and causes them to die. And when those dead plants decompose, it depletes the oxygen in these waterways, and that decimates aquatic ecosystems. So this is a major problem for water security in that algae can produce human toxins. And it's also a major problem for food security, given the knock-on effects on aquaculture. One fertilizer that's commonly used on agricultural land, not so much in the UK, but still very common globally, is poultry litter, which is the manure and bedding from poultry agriculture. So this and other livestock wastes are applied directly to agricultural land as fertilizer because they have a very high nutrient concentration. They also have the benefit of already being on the farm, so the transportation cost is low, and they're organic. So if you're trying to maintain organic certification of the crops, then this certainly fits the bill. However, there is a potential biohazard in that, though it's not very common, some animal pathogens can infect plants. And also the main point that I'm trying to make here is that animal waste fertilizers are not slow release. They break down in water. So the nutrients are immediately accessible to plants, and that means that if there's more nutrients than the plants can use, these will potentially be washed into waterways and cause nutrient pollution. What we can do to try to prevent this is to pyrolyze livestock waste to produce biochar before applying it to agricultural land. So pyrolysis is just heating up that feedstock in the absence of an oxidizing atmosphere, so in basically inert gas. Biochar has the benefits of being physically durable, so it doesn't break down in water, meaning that the nutrients are immobilized in soil. It's also chemically stable, so it leaches the nutrients slowly. Essentially, it acts as a slow-release fertilizer. And it's also known as a universal adsorbent because of its high surface area. So it has the potential not only to release nutrients slowly, but also to absorb excess nutrients that are in soil. However, it's important to note that there are differences in the properties of the biochar that you produce from different feedstocks. So biochars that are produced from animal waste are higher in inorganic, so they work more on the slow release fertilizer side, whereas woody or lignocellulosic waste produces biochars that are more porous, have a higher surface area, and therefore they're more on the adsorbent side of the equation. So overall, the idea here is that we take animal waste, in this case poultry litter biochars, and they will slowly release nutrients. Here we have nitrates, phosphates, and ammonium, which is what we're going to focus on in this study. And then the plants can use those nutrients, and whatever nutrients they don't use, the excesses would be absorbed by lignocellulosic biochars. So in order to test this, we wanted to simulate diffusion between buried biochar, in this case poultry litter biochar, and moisture in soil that contains soluble nutrients from added fertilizer that might ultimately enter waterways due to runoff. So what we did was we put these poultry litter biochars at a very low concentration into nutrient solutions at the maximum safe concentrations in the environment as determined by the Environment Agency in the UK. And we would shake these for 3 days at the average soil temperature in the UK, which is about 12 degrees Celsius. And we would see how the concentration of the nutrient solution changed. So we were trying to see whether we would get the expected leaching behavior, so the biochars slowly releasing these nutrients to the solution, or whether we would see them absorbing the nutrients from the solution, So that would imply that the concentration of those nutrients in the biochar was lower than the maximum safe environmental concentration, or whether we would see essentially regulation, which is oscillation around the safe range. And in reality, we saw a combination of these results. So for phosphates, we did see the expected leaching behavior. Over 72 hours, we saw about 100% increase in the concentration of the nutrient solution. However, in the case of ammonium, we saw that it was completely adsorbed within 48 hours. And finally, in the case of nitrates, we did see some regulation type behavior. So an initial leaching followed by adsorption, followed by another period of leaching. And this was within about 100% of the initial concentration to about 70% more than that. So in the case of phosphate and in the case of nitrate, we're getting to levels, we're either regulating or we're leaching to levels that are higher than the maximum safe environmental concentration. So we do need some help from lignocellulosic biochars to bring those concentrations back down to safe levels. So to test the performance of lignocellulosic biochars, we actually did more of an inline type of experiment as opposed to the batch experiments that we did with poultry litter biochars. So here we're simulating diffusion between biochars and actively flowing runoff. And the experiment looks like we take biomass, we put it into a custom-built pyrolysis reactor that sort of takes this cartridge that we can use as both the pyrolysis reactor and then ultimately the filter cartridge. So we take that, we heat it up in an inert gas, and we get this biochar in the same filter cartridge. We load a nutrient solution into that cartridge, and then we spin it in a centrifuge, in a microcentrifuge, to collect the filtrate, which we can then analyze for its nutrient concentration. So we repeat that process, adding fresh nutrient solution for each of 10 filtrations, and we can see that we do get some sort of regulation behavior below the maximum concentration, maximum safe concentration in the environment, unlike poultry litter biochars, where we found that we're regulating between about 100% and 170% of that maximum safe concentration. Here we're regulating between, for cocoa pod biochars, between 25% and 108%, and for coconut shell biochars between about 80% and 108%. So we can see that the lignocellulosic biochars are doing their job of keeping the concentrations of nitrates lower than the poultry litter biochars do. So looking back at this diagram, the initial idea was that poultry litter biochars would release the nutrients, and those that weren't used by plants would then be adsorbed by lignocellulosic biochars. However, we've seen that although poultry litter biochars are biased towards leaching and lignocellulosic biochars are biased towards adsorption, both biochars perform both functions. And in fact, in the case of ammonium, poultry litter biochars purely adsorb, and therefore it would be necessary to add ammonium in the form of conventional fertilizer to soils in addition to this biochar regulation system. So far, we've shown that poultry litter biochars gradually leach phosphates over the 72-hour time period that we tested, so they do have that slow release fertilizer function that we were expecting to see. They also regulate very high concentrations of nitrates. So they leach some nitrates, but then when the concentrations get very high, about 70% higher than the maximum safe concentration in the environment, they can bring the concentration of nitrates back down to that maximum safe level. And we also saw that they actually completely adsorb the maximum safe concentration of ammonium within just 48 hours. We also saw that lignocellulosic biochars were able to regulate nitrate concentrations down to below safe levels. So if you have poultry litter biochars that are slowly releasing nitrates for use by plants, but plants can't use all of those nitrates, those poultry litter biochars, if the concentration gets high enough, will adsorb them back down to the maximum safe levels, and then lignocellulosic biochars will take over and regulate them back down to even lower levels, which overall drastically reduces the risk of nutrient pollution of waterways from agriculture. So these are just preliminary results. There's still lots to do. We want to make sure that we do both batch and inline experiments for both feedstocks, all feedstocks, for both poultry litter biochar and the lignocellulosic biochar feedstocks. We also want to perform these experiments in water, so we can see if there's leaching behavior when the concentrations are much lower. So for example, we might expect that even though poultry litter biochars were only shown to adsorb ammonium, if the concentration was sufficiently low, if there was no ammonium in the solution, would they in fact leach some ammonium? And that leads into the next point, which is that we want to extend the duration of these experiments because it's possible that if we had left the solution that poultry litter biochars had adsorbed the ammonium from for longer, those adsorbed, the adsorbed ammonium would eventually have been desorbed after a sufficient period of time. And ultimately what we're trying to do is to determine ratios of poultry litter to lignocellulosic biochars that will regulate concentrations of nutrients in soil to safe levels long term. Thank you so much for listening. First of all, I'd like to thank the students that completed this research. First of all, Ahmed, who's a master's student in my lab, and two interns, Elena from MIT and Punia from the University of Surrey. I'd like to thank the funders, who include London South Bank University, UKRI's Research England, and the Office for Student via South Bank Innovation. I'd also like to thank Toxins, who provided the lignocellulosic biochar samples, and Inspro, who provided the poultry litter feedstock, as well as the Environmental Biotechnology Network, who paid for my travel here via their early career researcher travel bursary scheme. And I'd like to encourage all of you to get involved in the working group that I coordinate for EBNet, which is the Biochars for Pollution Prevention Working Group. And you can get involved by either sending me an email or scanning the QR code, which will bring you to our working group website. Thanks again for your time.